Hello everyone, my name is HyoungsooKim from KAIST. Today I will talk about direct measurements of fluorescent emission. So this work is collaborated with the biological science team. So today contents are following in this way. So we can easily smell fluorescent everywhere. However, have you ever thought about can we see directly the fluorescence from the flower? So if we can, why it is so important to see this signal? As you may know, if you're planting the seed, so they're gonna grow and then eventually they have a flower and fruits. However, to get a fruit, it is more important to have some pollination process, but which is done by the third party, actually done by insects. So in fact, it is well known the fragrance of flowers affect the interaction with the pollinators, microorganisms, and some insects. For instance, many flowering plants can tune their scent emission rates when pollinators are active for pollinations. Thus, visualizing scent emission can help us to understand the ecological evolution of plant-pollinator interactions. So due to this reason, many groups have been trying to develop a method for scent analysis. So in general, typically they collect the fluorescent from the plant or flower, and then they use the mass spectrometry to quantify or to measure some kind of quality of the fluorescence. But by using this kind of conventional method, it is impossible to directly measure the releasing frequency from the flower. So recently, people tried to invent or develop some new measurement techniques. For instance, we're developing some kind of the potable measurement, and also the smartphone or the detection system using chemo-responsive dyes have been used to measure volatile organic compounds, which is the fluorescence. However, still it is impossible to directly measure the time-dependent emission rate of fluorescence. So this slide is showing you what kind of different types of the gas sensing measurement techniques in the world. But there's a bunch of different ways. However, it is still tricky to directly visualize the fluorescence in time. However, among the different types of the measurements, so optical interferometry might be the best way to directly measure the fluorescence because it is available for real-time measurements and it has a good spatial resolutions, and also the sensitivity is a kind of reasonable range. So interferometry techniques are described in this slide. So typically using one single laser, and then they're gonna split two beam line using beam splitter. So one beam is going through the void fraction, which is the reference, and the other beam is going through the measurement domain. So in this place, we're gonna install the flower eventually, but let's assume some volatile liquid is evaporating in the air. So eventually, you're gonna interfere two different laser beam, and then you're gonna record it. So here I can show you without any kind of the volatile liquid, so basically no sample case, you will have totally straight fringe patterns. However, if you have some volatile liquid in the measurement domain case, you will see this kind of distorted beam line. So before applying the interferometry techniques to directly visualize the fluorescence, so we should know what kind of the volatile organic compounds will exist in specific flower. So for the flower, we select the Really. So Really is one of the most strongest fluorescence emit. So we use the gas chromatography and then we analyze the components. So we collect the fluorescence from the Really using conventional way, and then we use the gas chromatography mass spectrometer, and then we analyze the compounds, and we found out that Really contains 10 different chemicals. Among them, the linalool is the most dominant chemicals, and which is the volatile organic compounds. So basically, the setup looks like this, and then we install the flower in the measurement target here, green box. to collect the fluorescence, we prepare those kind of the confined chamber, and then we are expecting if they really emit the fluorescence, then they're gonna accumulating from the bottom side. And we investigate linalool has this kind of the molecular weights, and also it has this kind of the refractive index difference, and it has a big difference between or the airs and the linalools. So we are expecting we can see some kind of signals. So this is the first experiment. So every 5 minutes we collect a certain amount of the release of fluorescence, and then we measure the fringe patterns, and then we analyze it. So you can see here phi is kind of the relative amount of the fluorescence which is confined in the measurement box. So we observe that as time goes, the signal is getting increased. And then after that experiment, we clean up and then re-preparing the chamber, and then we do measure again, and then we repeat this kind of experiments every 5 minutes, and we collect all the signals. And right hand side, we compare the relativity of the signals. So this phi is plotted in the different times, and you can see at the beginning point there's nothing about the signal, but as time goes, along the y directions, which is the vertical directions, you can see a lot of the fluorescence are accumulating from the bottom side. So this signal tells you in time really emit certain amount of the fluorescence. So we want to measure the longer time measurements. So we prepare the 5 different lily samples in random way, and we change the camera which is able to measure the longer time, so we can measure the signal up to 90 minutes continuously, and this is the results. So we prepare the 5 different samples and then continuously measuring those kind of relativity of the signal from the fringe patterns, how much distorted the fringe pattern signals. And here you can see the results. All different release have a different profile of the signals, which means this continuously really emit the fluorescence. So based on what we measure the signal from the top signal, we calculate the FFT or the spectrum. So we obtain the different emission frequency. So the first has a 21 minutes, and second and third has a 8 minutes, and fourth has a 14 minutes, and last one is a 50 minutes. So depending on the lily, so we observe the different emission frequency. Based on the previous measurement results, we can also do some kind of the temporal spectrum measurement analysis. So based on the previous measurement results, we replot the data in different times with a different segment of the period. But here you can see the signals. So the temporal spectrum profile showing you the blue signal, it means there's nothing, but the red signal means that there's a strong fluorescent signals. So here you can see, well, depending on the flower, you have a different the signals. So based on this kind of the laser interferometry measurement techniques, we confirm that really flowers emit the volatile compounds discontinuously with the pulses observed around every 10 to 15 minutes. Thank you for your attention.